these mechanistic models of something like this part here, with a chemostat. And this chemostat, I also mentioned this, but I want to emphasize, I didn't realize how useful it was when I was a student, and it's in the sense that I think I mentioned there are people that model the abundance of microbes in the ocean around the whole planet. I wish I had put here one of these pictures, the amazing simulations where you see the phytoplankton growing, seasonally, and then the cycling of organic matter in the whole ocean. They use this thing. They just have one meta species that is called heterotrophic microorganisms that would consume organic matter, for example. And for each, they have a volume that is the ocean, and there's partition in little blocks, and in each block there is a chemostat. That's where they integrate, where there is a flow of resources in, a flow out. It's at equilibrium, so these flows are the same, and these things can grow. That's the ecological dynamics. So this is what they use. So in that sense, it's pretty useful. And then I told you also that maybe you can go down until here. But going beyond that is very difficult. It becomes very impractical. That's the issue. It becomes impractical. And for instance, I told you, two things that are important here. You can have the situation where you have a resource that feeds a species, it excretes something, for example, a fermentation product. And that is the food for somebody else. But this fermentation product inhibits the growth of the first one. I can give you an example. Well, it doesn't matter too much, but for example, cellulose degradation, really important, something that in fact in Brazil people do quite a bit of research in how to convert cellulose into fuels. And this is one of the main issues, that the organisms that degrade the cellulose, they ferment for reasons that are not important here. They produce, this releases hydrogen, and hydrogen inhibits their growth. And there comes a methanogen, that is this guy here that removes the hydrogen, and now this guy is happy. But that's a very simplified system. There are more species and so on. But then modeling this thing becomes now you have to resort to some more qualitative phenomenology, which is, could be fine for the purposes. But then doing this type of things where you're measuring parameters very detailed, it becomes difficult here. And now if you start to expand to large systems, impossible. So now we have to do the type of things that, for example, Stefan have been talking about. So that's my message. And just one more thing. When you have these feedback loops, which, by the way, I want to also make clear, because I will talk about this, I think the systems that I study, and I will discuss them, typically have this structure, not many feedback loops, although we have not quantified that systematically. In anaerobic systems, I want to be explicit, this is very common. And one of the main reasons is the bioenergetics. When the delta G approaches 0, the accumulation of this thing, R2, the product of the reaction, inhibits the reaction from moving forward. It's thermodynamics. And so that's why you have these things. And when you have those things, when you have these loops, now the dynamics of the system can be very rich. And nobody knows how to model this, but you can imagine, some of you can imagine, first of all, if this inhibits, then you can have a lot of coexistence because they self-limit, right? If you have another guy here that is consuming this, let's call this 3 and 4, you can have multi-stability. You can find models very easily that give you multi-stability. Because this becomes like a cooperative system, another cooperative system. It depends, and your initial condition determines where you end. Okay, but modeling these things, I mean, we can make all models we want, but the data does not exist. That's the main issue here. So we can play games with toy models, but there is no data. Okay, that's my summary of last lecture. And so today I'm going to talk mostly, or really, the rest of the lecture is all about experiments that we have done in the lab. And one of the I want to start with this experiment. Sorry. Yeah.
So you say that there are no data, but there are models. But are there models that take into account, like, seriously the thermodynamic constraints?
Yes.
Okay.
Yes, there are models that include that would.
But that like combine the two things, right? They combine the
Right.
thermodynamic constraints with the population dynamics that emerge from them.
Yes. So basically, you have other types of, like, instead of having monod kinetics, this monod kinetics comes with factors that depend on the delta G. Yeah. That's, it can be done. Whether that's the right way of doing it or not, I'm not sure. And it's, but I have not seen this done in a large scale. It's like here, look, we can derive this formula and apply it for a small system, but then validating experimentally and so on, I don't think we are there yet. And traditionally, like when people used to do this back in the, whatever, 60s, 70s, they did not include a specific function from thermodynamics. It was just like whatever decaying function from, yeah. And okay, I almost forgot to say, I sent an email to Tiago, the guy with the media, and so you, I don't know if you already got it, but you will get some link with some resources. So one of them is this thing. This is it's an art program. I don't know, it's a Shiny app. You can make these interactive applications with R, and I made one for the chemostat. So you can see now the phase space there, and you can play with this, change parameters, and see what happens. So if you make, I think for those of you that like mathematical modeling, the model I wrote it last time. You can play with it. You can change it, get some intuition, and you have here the phase space. You can check that when you draw the phase space, this works. And I think if you're analyzing small dimensions, low-dimensional dynamical systems, I mean, for sure, you have to look at the phase portrait, and then you know everything. So it's a good way to work. Another resource along those lines, maybe more in general, useful for those of you who like mathematical biology, comes from this guy here, who was he was my teacher when I was in Netherlands. He's a really good teacher. And he also he does a really good service for the community. He developed one of the first programs to do phase portraits and analysis of dynamical systems. And now they translated it to R. So there is a really nice R package that you can download and get these super nice visualizations of the dynamics. This is a Lotka-Volterra system, and you can look at the phase portrait. Obviously for low-dimensional things, this works. So it may be useful for some of the students. That's why I wanted to share that. And there are also two papers that I shared, one on the bioenergetics and the other one that I mentioned last time with the simulations of the trophic systems. Okay, so that's that. Now, experiments. So, this will happen. This just takes a while. Maybe I want to say something unrelated. Now, okay, I started already, so I'm going to say it. So like I told you already before, I am from Ecuador, and I did my master's and PhD in the Netherlands. How many of you, students, I guess, would like to do a PhD, postdoc abroad? Let's say, yeah. I imagine most of you. So for whatever reasons, historical, I don't know, most of the science, most of the money, I guess, for science is in places like Germany, Holland, the US, the UK. In all these places, people really dislike when you are not on time. I learned that the hard way. So it's a more serious problem than you think. So, think about it. People really want you to be dependable and all these annoying things. So I don't mind. But, well, I mind, but not now. But I'm just telling you because these things matter. All right, so here we go. So with experiments. So how do we measure community structure, community dynamics, and so on? So first, how do we get measurements? And Jacopo mentioned some of the things. I'll tell you a little bit about the techniques. That's not super important, but just to be thorough. And then we're going to go to the interesting questions. What are the dynamics of community assembly as measured by experiments? How do they compare, for example, the things that Stefano was talking about? So we'll see that it's totally different. Which is not true. And then we're going to go to this really, to me, important philosophical question, but but scientifically very important, which is what are the right variables that we should want to measure? And how does it relate with what we measure? Which I think also a good topic for a roundtable discussion. Okay, so it turns out that we do what technology allows us. And this relates to the last question in my previous slide. And so some of you already know very well, for the case of microbes, this is one of the main advantages is that we can use this next generation sequencing techniques. So we can extract the DNA. Basically, for those of you that don't know, you basically crack the cell open, the DNA comes out, DNA is a big molecule, then you fragment it into little pieces, and you put adapters around. These adapters are other pieces of DNA. This is the magic of molecular biology. They can do all these games of pasting things together and so on. And these adapters will go and bind to these things called flow cells in a sequencing machine. They will bind there. This all happens at, you can imagine, like super small scales. And they start to get amplified by a chemical or a reaction called the PCR that is happening at these super small scales. And this emits light, and these machines read the light and know what are the sequences of these little pieces that are being reproduced in these flow cells. That's a very clumsy explanation, but that's more or less what happens. Now, the important thing here is that you can do that with any piece of DNA. And so, for example, in my lab, we do a lot of this type of sequencing, but where we sequence the whole genome, all genomes. We try to sequence all genomes completely in a community. These communities are small. So we can get all the DNA from the community, fragmented, and all these little pieces are going there. They're being amplified, then you get the DNA, and then bioinformatically in the computer, you try to put it back together and reconstruct the genome. Okay? That's something that it's quite nice to do. Then the more common approach, cheaper, and it's what the type of data that Jacopo was talking about, is to forget about most of the genome and just focus on one little piece, which is connected to the ribosome. And so you amplify that little piece, but then you basically do the same. You put these adapters around it. Okay, so it's a bunch of steps there that are not super important, but you put these adapters around it, you amplify it in the machine, and then you get your data. The only advantage of this, and why it was a revolution, in the sense that now we have so much data compared to, for example, 15 years ago, 10 years ago, is that you can mix all you say, for example, I have I take a sample from my saliva, extract the DNA, amplify only that piece that is connected to the ribosome, which is this ribosomal 16S. I'm going to mention maybe these words again, 16S, for those of you that know. And then I can just sequence all of that, and then later reconstruct the sort of the species that were in the system. So I don't need to separate them. I can just extract everything, and then this gives me an estimate for the species. So, okay, and I'm not going to really say why the 16S and not other regions. Turns out to be a reasonably good marker for these things. Okay. What's happening here? Yeah. Yes, thank you. What's going on? Thank you. Sorry. Actually, I should mention that, okay, this 16S is connected to the ribosome. The ribosome is super well conserved evolutionarily across organisms, right? We all have ribosomes, otherwise we cannot make proteins and so on. So you can take the sequences and build a tree of life. That's how the tree of life is built for bacteria in most cases, okay? So that's one statement. So you can sequence your community from my saliva, for example, and I'll project it back on a tree. And so you can, if these slides would move, you would see, yeah, so you can project it on a tree. So that thing on the left is a phylogenetic tree that more or less describes the evolutionary relationship between your species that you have sequenced. And for example, on the right, this is just an example I found in the literature. You can look at the abundances of each of these species, but you can also see how related they are. Okay, so that's kind of that's basically the structure of the data that you get here. And then the rest is whatever, statistics, whatever you want to do with this type of thing. But that's the raw data, more or less. Okay, so now I can tell you about the experiments that we have done. Sorry. Okay. This person there is Leonora Bittleston. She was a postdoc in my lab. And she did this type of, she actually is an expert in these things called pitcher plants, which are, who knows what's a pitcher plant, by the way? You know. It's a carnivorous plant. And actually, I think she did work in Brazil as well, because there are some of them here. These are beautiful systems for community ecology because each of these plants, so they have like a pitcher, that pitcher with water, that thing there looks similar actually, just smaller. And it's filled with fluid from the rain. The microbes are there, and the poor insects that fall in there, they get degraded by the microbes. And that's what the plant eats. And so we were interested in using the systems to study community assembly. And so what we did was, what Leonora did, was she went to a field close to MIT, well, close to Boston, in Harvard Forest. And she collected the samples from the plants, brought them to the lab, and did this type of experiment that you may see frequently in the literature nowadays, which is sort of a selection experiment where you put in some sample of the initial inoculum from the plant, you let it grow for a period of time, then you dilute it, and the whole thing repeats again. Now it grows. And now you keep passing it. You keep serially passaging this. Okay? Is that clear how that works? It's what's illustrated here. So initial volume, we let it grow for 3 days. Then we have to dilute it. This is diluted in something that sort of mimics rainwater. It's just water, a little bit acidified. And then we let it grow for 3 days, dilute it, let it grow for 3 days, dilute it, and so on. This went on for a long time, for like 2 months. And then it's a different project where we kept going for like 6 months just to see how this would evolve. Now I'm talking about really mutations and so on. But that's not what I'm talking about now, just the first assembly part. Okay, so I'm going to show you the next slide. It's going to be really packed with data. So we're going to spend some time trying to parse out. By the way, ask me questions if this is not clear at any point. Okay, so we had 10 different plants, 10 different pictures from this ecosystem. And so each of them went through the same treatment, okay? So what are we seeing here? The top is how much DNA is were we able to recover after each of these passages. And you see this generally increases, okay? So initially there were low abundance of these microbes, and as you keep going on and on and on, this went for 60 transfers. Here's the number I was forgetting, for 60 transfers. It's like 3 months. Then, this kept growing in abundance in biomass. Fine. Now, what we have here is the species composition, so to speak. But you will see these names again. It's also what Jacopo was mentioning. These things are called ASVs. And these are just some type of clustering of the sequences. Before, people talked about OTUs, operational taxonomic units. This is a new standard in the field that has an error correction model. But it basically, it's our best approximation for the different sequence types that exist in the system. That's maybe a better name, ribotypes, rather than species. Who knows what's species? So the different things that we can count in the system, okay? And their dynamics are here in different colors. Right. A lot of things happen. They have the dynamics. They more or less by eye seems to stabilize and so on. But they're different. Each of these things are different. That's one statement it says. Here are the names of the things. We cannot make much sense of this. I know, even if you're a biologist that knows about microbes, this doesn't tell you anything. These are Latin names. Fine. Now we can look at these dynamics in kind of a PCA, non-dimensional scaling, and what you see is that they start, this is where they start, in these points there, far from each other. They more or less converge somewhere, but not completely. They remain different. And then, so for example, you just look at this microcosm 4, MO4, starts here, quickly moves to this space here, and then just wiggles there. This one stays here, another one stays wiggling here, and so on. They reach somehow like kind of an equilibrium that is not exactly the same from each other. So, and that's what you can see here, which is, this is what we're plotting here is over time, the dissimilarity between adjacent time points. So initially, so after the first transfer, there was a lot of changes, and this number of changes keeps decreasing and decreasing as you approach some time, which was like 21 days, where it looks like more or less stabilized. So this is also what Stefano was saying. We could have stopped somewhere here, and you know, we didn't need to do all this. We didn't learn much more from this last part. And finally, I guess this is kind of interesting too. So you can look at, for each of these ASVs, these ribotypes, species, whatever you want to call them, you can look at what is their multiplicity across these microcosms, meaning are they present only in 1, in 2, in 3, in 4, etc. That's the histogram of that questions, that answers that question. So most of them are present in only 1. This is at any given point. And then only a few are present everywhere. And then this is somewhat correlated with the relative abundance too. Those that are present everywhere tend to be abundant, okay? And those that are present only in 1 tend to be low abundance. That all makes sense. Now I guess this is the interesting part. Exactly the part where Matteo is leaving. No, it's okay. he had a rough night. So this is right. So the richness. I heard someone ask the other day, what is richness? It's just the number of species. Okay, the number of these things that we can count. And these are the different communities, the different microcosms. As you can see, these things decrease in time, but very consistent with what I said, right? You have this process. By the way, I should say maybe, so you come with a lot of species from nature, and you put in these conditions where you're asking them to grow. Oh, I didn't tell you this, by the way. It was, they were growing on, we have to provide a carbon source. So I didn't say that. You should have asked me. So, and we provide these ground insects. So we take dried insects, you can buy them on Amazon, and they're already like a powder, you put them there. So basically, it's kind of, we're trying to replicate the pitcher plant, you know? And so we are transferring them, putting these insects and so on. But still, it's not like the environment. For example, there is no migration in. That's one big thing that is completely stopped here, right? Plus, who knows what else? Maybe the plant also provides some things that we're not putting in here. It's just a glass tube with the water, the ground insects, and the microbes. So of course, you have this process of species loss until they stabilize. But each of them stabilize at a different point. Some of them have a relative high richness, actually close to 100, and some 50, and so on. It's a pretty large number of species for a synthetic system. Then, this is the thing that relates directly to what Stefano said the other day. This is the random zoo experiment, okay? And in the random zoo experiment, if I understand correctly, you know, it doesn't really, the effects of the interactions are negligible. So if you start with n number of species, even though they may interact in different ways, what you get out in terms of who can coexist at the end of this assembly dynamics is a proportion of how many things you start with, right? And that's exactly what we see here. So, and I was really puzzled by this, actually, because, right, so for example, this guy, MO9, this particular community, at an initial time point, which we will discuss, by the way, is not time 0, it's day 3. It has a low number of species, and at the end, it's pretty much it's proportional, really. And compared to, for example, the 6 or 2 or whatever, which have a high number of species, and they end up also with a high number of species. So more specifically, these guys here, for example, have like 100 species at day 3, end up with whatever, 45, 50 something. So about half. They lose about half. That's the thing, regardless of anything else. So I was really puzzled because, you know, the only model I could think of that would explain this is when there are no interactions. right? And but then right, so that what you mentioned explains this. So that's nice because it's a nice consistency with the theory. Right. Then what else do we have? I don't know. Yeah. Good. So now we looked at the probability of extinction of a species in each of these systems. Turns out what you can see there is kind of noisy. But interestingly, this black line here is not a fit. What you have here, these black points, these are the averages of each of the systems with their standard deviations. Okay, and the black line is a model that is based on a geometric probability distribution of extinction. And this model is like this. It has only one parameter, which is that probability of extinction. Okay, at each serial passage, there is a fixed probability of extinction according to this model, and that's the only parameter. And you can estimate this from the data. And then, so there's not really a fit. It's just what you put in there, and then and this is the best model that explains the data. So it means simply that it seems that for all these different communities, regardless of their species differences and whatnot, they have the same dynamics that are explained by one parameter, which is the probability of extinction, which is independent of the system.
Just to be sure, so what you are, so to get the black line, you don't need any fit because you are using the red line over there.
Yeah, you're using the mean time to extinction from the thing.
But I mean, also there, if you take the integral over that over time, right, that is the probability that a species go extinct at any point in time. So
yes.
that integral should be 1/2.
Yeah, I suppose, yes. Yes. Mm.
Just to
Yeah. Okay. Okay, and the last, I keep pressing this, but I don't have the. And the last thing here is you can take your richness over time. If you normalize it by the initial richness, those decay curves that are shown here in the first panel, they all collapse. It's all the same. Yeah? So because this follows also from the other observation. So basically, all the systems behave the same in all ways, regardless of their idiosyncratic differences. So that's kind of nice. Especially, I guess, for those of you that are physicists, you may find that appealing. Good. So here's the other aspect of this data that I wanted to mention. So we can ask this question. Basically, your species identity, or really the fact that you have that sequence, how much does it determine your dynamics? So this same sequence, the same species, in a different context, so therefore a different microcosm, will it have the same dynamics? Yes or no, and to what extent? That's the question. Okay? The answer is by and large, no. So these are, the distribution here is the distribution of correlation coefficients between the dynamics of the same species in two replicates. That's the distribution that it picks at one. It replicates, okay? So it should pick at one. So that's okay. But there's still a tail somewhere there. In some cases, they are anti-correlated. But okay, fine. In most cases, it's well correlated. That's great. If you would pick two different species in any of the systems, the distribution of correlations is pretty much centered around 0. So they are not correlated at all. That's fine. That also makes sense. Now, if you pick the same species in two different contexts, two different communities, is not too different from the one in which you pick two different species. It's like it's a little bit shifted towards the right. There are more positive correlations, but there's really not much going on. Okay, so the context where species exists matters a lot for its dynamics. Despite of what I told you before, that when you look at just richness and so on, everything looks the same. When you look at individual species, it can look very, very different, okay? Perhaps, I don't know, this one I thought, I don't really know, but perhaps this is the kind of thing that we can consider as environmental noise. The same species with the huge fluctuations when you look across different systems, where the only difference in this particular case is the biotic context in which they exist. This, I don't know if you will find this, yes.
Did you try to do this? Did you try to do the same graph only for the abundant species, right? Because the other ones would have more fluctuations in these correlations.
Yeah, I'm not sure if we have that systematically. I can show you. Indirectly, we may be able to see that in one of the next figures, but it's a good point. But although actually related to that, you can, and so this is, this is actually important for those of you that know phylogeny and taxonomy, I guess the biologist, you know that you can have like, I think I have it written somewhere here, but you can have, the species, then you can group things in genera, like, it was a good example, like I guess Homo sapiens, and then Homo is the genus, Homo sapiens is the species, and then you have family and classes. So you can group things at really higher taxonomic levels, which should, if for example these problems of the low abundance species should reduce because now you are grouping things, you have less variables, they are more abundant, and so, but the same result if you group things at a family level, which is pretty high, for the biologist, it's like saying pseudomonas, but there are like millions of different pseudomonas. They're all in one thing, and you still see the same thing. So this game of grouping things at different taxonomic levels here, it doesn't really help. And this is another view of the same data. Here we're looking at all these different ASVs, different taxa, and the dots indicate. For example, let me pick one nice example. Let's say, well, I know it's hard to see, but let's say this one here. I hope you can see the pointer. So there were 10 different microcosms, 10 different communities. We gave them different colors. If you're colorblind, I'm sorry, I know this is terrible. It's a terrible color scheme, but whatever. So you were first detected here. That's what this means. First detected here. And for this particular case, last detected at the end. I can see a blue color here, which means that it didn't go extinct. In some cases, though, for example, we're still with this one, right? In the orange one, it was first detected here, also existed. Actually, in this one, it existed until the end in all cases. But there are some cases where you had the dots before the end. That means that it went extinct before the end. So we detected it, and then it went extinct. Detected, went extinct, and so on. And you can see basically the spread here is huge, which means that in some cases it goes extinct at the beginning, also in other cases at the end, and so on. It's really messy. And these are just specific examples of these dynamics. So and this is a guy that is really abundant, right? It goes to 30% of the community here. In a different microcosm, it goes up and then declines and it goes extinct. And in others, stay there at low abundance and so on. In all these cases, it can be very different. Here it goes there, here it goes extinct, whatever. So again, it's just the same point that dynamics are correlated, are really different for the specific taxa when they are in different communities. I think, yes, please, yeah.
I just have a question about the experimental setup.
Yeah.
How are the initial conditions in these different replicates, I guess? So are the relative abundance of each ASV similar from in all the replicates in the beginning?
No, not necessarily. Not necessarily. No, we don't know that. Yeah, right.
Okay.
So yeah. So it could be there is an effect of the initial densities, for example. Although, well, I mean,
but
it has to be more.
Are they all really low then from the beginning?
They're all very low.
Yeah. Okay.
They're all very low. Yeah. Basically, some go up and then go down, and some others go up and keep going up and stay there. So, yeah. But that's a good point. Oh, I forgot to say also. I forgot to say that this thing here, we did this from day 3. If you do it from day 0, then you don't see anything, right? So that's kind of interesting too, because at day 0 it's just what's in this in the wild, basically. There's the scoop of the wild. Most of these things, the vast majority, are not going to be able to grow. They are not adapted to this. They're just going to die immediately. After day 3, you have already gone through that first selection of what will persist, and then from then on, it's just dynamics of growth and competition and whatever. Yeah.
How big are these?
Sorry, how big are these plants in terms of, and do you take all the sample, all the water from the?
No, we took we took 15 mils of the liquid.
Not from the bottom, or you can have all circulation in the plant?
No, these are pretty stagnant, so when you look at the plants, there is a lot of these detritus from the dead insects and so at the bottom.
And you don't take that part?
No, I think we mix it and then filter it.
Okay, you first mix it.
Because there are a lot of predators, protists.
Yeah. Okay.
So okay, I think I made my point here. So I'm gonna skip this one and leave you with these two. Oops, these two. These are the main points of this part, which is something I said already, but just to reinforce it. It's the same point that Stefano made before. It seems, at least at the scale of this system, very diverse and so on, that these interactions did not matter that much in terms of the number of species that end up coexisting. Well, I mean, you saw quantitatively what's the statement. They are directly proportional to what was able to enter the system at day 3. That determines what stays after 63 days. And so that is consistent with a model of no interactions, which we know cannot be true, because the species have different dynamics in different systems, so interactions must play a role. It's just somehow it averages out, I don't know, maybe perhaps the way you described. Fine. So, and then, like I said, the dynamics of species are context dependent, and in this case, it's the biotic context, not the abiotic context, which was perfectly controlled. All right, so now I'm going to switch gears a bit, and I'm going to tell you about what actually is most of what we do in the lab is based on the system I'm going to describe next. I have probably like half an hour, no? Or yeah, that's good. So the purpose of this is to tell you, the nature of the system, the things that we have found. Just I'm going to highlight a few. And the main philosophical point that I would like to discuss with you, and in the next lecture I'm gonna try to address it. How we're trying to solve that problem is what are the relevant functional units of a community. I can tell you the bottom line is that I think we are measuring the wrong things from the beginning. I don't understand. I mean, it could be an interesting thing to look at. Why is it that we are so obsessed with species when we don't even know what's a species, yeah? So, and then we end up counting species. And in this particular case, in microbiology, the technology is what the technology forces you to do in a way. So, but these are not the right variables. And we are at this point I think in the field where the challenge is how to define the right variables and how to learn from the data what are the right variables. That's going to be really the big message that I also want to talk about next time. So, this is the ecosystem context where our work sort of begins. That thing you have there, I'm sure you know, this is an algal bloom. This is the coast in front of France and England. This thing is huge. It's probably bigger than Ecuador. And there's a huge amount of organic matter of carbon that enters from the atmosphere to the ocean by primary production, okay? So this is incredibly important for the planet. Now, what happens with this thing? These organisms, these algae, these are coccolithophores, but that doesn't matter. They die, they excrete things, and then some of the things are heavy and they are non-soluble, like the shells of the little algae or the copepods that eat them, they also die, and they poop, and this organic matter starts to sink, okay? And oceanographers have been doing this for many years. This is 1980s, the next slide. And they put these traps on the water column that measure how much they can trap organic matter sinking for a period of time. So they can calculate a flux down the water column. And these things look like this. This is the original paper from this guy Martin. This is called the Martin curve. People try to do a lot of math with this, But it turns out you can fit anything. You can fit an exponential, you can fit a power law, and the data is not good enough to distinguish. So we're a bit stuck with this. Well, not we. I don't really. It's not. I don't work on these things, to be honest. But oceanographers have a lot of models, but this is just the data that exists. But it basically tells you that here there's a lot of particles, a lot of flux of organic matter at, say, 100 meters. And if you go down by whatever, 500 meters or so on, you have lost most of this organic matter. Presumably because the bacteria have degraded it, presumably. It could also be that it got eaten by something else, I don't know. That it became less heavy, became buoyant, I don't know. But the idea at least is that the bacteria are degrading these things. So what you're measuring here is the digestion by microorganisms, okay? But what's appealing to me is that this has clearly two physical scales. One is the tiny scale of these little particles that are sinking, and the other one is this big scale that people are measuring here. So now I'm interested in this small scale because that's where I think the community ecology emerges. And so to give you a cartoon vision of how how I started thinking about this at least. You have these particles, right? They're of course not perfect spheres, but I guess if you're a physicist, you're fine with this. And then the microbes are around. They are in the water column. And so this is the pool of species that will colonize your patch. It's like a meta-community. And then they will attach, they will grow. You can find these bacteria have specific adaptations to adhere to the organic matter on the particles. Then they start releasing enzymes. This is actually going to be critical for the last lecture. They start releasing enzymes that digest organic matter. So that releases the sugars. I mentioned this last time. This step here, this is complex organic matter, and there's a species that utilizes. This is what they actually do is they digest it, and that releases sugars. This happens outside the cells. This happens outside the cells, which opens all these public goods dynamics, which I will talk about later, but I mean, in another lecture. Basically, these are public goods, all these sugars. Somebody else can use them as well. They don't have the rights to use them only. So it's all determined by diffusion from there on. And then there are also other metabolites, like the R2 down in that picture, that are released. And that invites another species to grow and so on. And then you have the assembly of a trophic system. So that's the concept. And ultimately, these things are consumed. And the bacteria need to leave and find a new particle. So that's the game they play. One second. Oh, here it is. That's what I'm looking for. Here. No. It doesn't work. Okay, too bad. All right. You know how to make this work? This is USB. That's HDMI. Beautiful. Okay.
But that's the yesterday I had that.
Huh? No. Oh, too bad.
Same problem yesterday.
Oh, that's matter. Okay.
I will check.
Yeah. Anyway, so I think ultimately the game that these organisms play is like colonizing a patch, degrading it, migrating, finding a new one, and so on. And in each of these patches, there is this process of community assembly. So I wanted to know what's going on with that community assembly. How does that work? And to be perfectly honest, I started doing this thinking I wanted to study social interactions, those public good dynamics that are supposed to emerge because of the release of the sugars. I was supposed to find cheaters, these organisms that would take advantage of sugars but not produce the public good and so on. That became really hard to do. Now we know how to do it, but that's not what we found. So experimentally, this is how we do things. We use these synthetic particles. We don't use the real particles. These are beads made of hydrogels. In this case, the hydrogel of chitin. And that black thing there is a magnetic core. And so that's 50 microns. It's very small. And then we put these particles in the sample in seawater, just raw seawater. And they get colonized by microorganisms. which are stained there so you can see this fluorescent green thing is the bacteria attached. Then you can sequence with a 16S, for example, and look at the dynamics. For example, maybe it looks like that. That's a sketch. Then we can cultivate the microbes in the lab so we can study their physiology. Maybe do those measurements like with the chemostat and see what is their affinity to the sugar, how fast do they grow, and see if that helps us understand anything. And we can map these microbes to their dynamics in the communities. So then we can connect the physiology with the dynamics of the community, which is, I think, ultimately one of the things we like to do. And the kind of games that we're interested in playing now is assembling synthetic communities and seeing if we can predict anything from the genomes or that type of thing. So that's kind of our platform to play in the lab. And so this initial experiment, like I said, you put these particles in a bottle of seawater. They are rotating just to keep them agitated because the particles are heavy, otherwise it would sink. So we keep them rotating there. And then we can sample them because they're magnetic. And so we sample about, I think about 1,000 particles per time point. And one of the advantages, so what's happening here is that this is seawater, okay? And we drop in 1 million particles per liter. And so now every particle is being colonized, presumably at some rate. You know? And then we sample about 1,000. So then the data we get is an average of the community composition across 1,000 particles or something like that. Let's try this.
If not, it's probably that is
Yeah. Veamos. No. Oh, ¿me quieres que prenderlo? No. No. Este tampoco funciona. ¿El qué? No. Oh, quizás porque lo apagué, pero bueno, no importa. What happened now? Okay. All right. Anyway, so we get this. We sample. This is annoying. We sample over time, and then we sequence. Then we can measure the community composition over time. Okay. Everything clear so far? Yes. How we do this? So this is the data that we get. So what you're seeing here is I don't know why its letters are so big, but this colorful thing here is a heat map, okay? And the rows of the heat map are the different species, the different taxa. The columns are time, okay? And these rows are normalized, so they sum up to 1 each row. So the red is maximum abundance, and black is absence, okay? But it's important that they are normalized like that. Otherwise, you wouldn't see anything for the initial time points because over time, biomass is increasing. So the things that are at the end are really much more abundant than the things that are at the beginning. That's why we have to normalize by row. Okay? Now, we had three replicates of this, just 3 different bottles. And turns out, if you look in this case, which is in a way, now that I think about it, different from what I just told you about with the pitcher plants, you look at one of these particular taxa, and the dynamics are really reproducible across the different successions. Because these are successions. You know, you start somewhere, species grows, then dies, and in the meantime, another comes, grows, dies, and it's a sequential process of invasion and growth. And the dynamics are really reproducible. All right. Then, you know, we had a sort of a slice of this matrix are the things that we have in culture that we can investigate with experiments in the lab. And so that's that slice there. Okay. Then for those things, we can ask the questions that we think make sense to ask, which are, well, remember those particles, those gels were made of chitin. Chitin, by the way, I don't know if you know, in Portuguese must be like quitina, like in Spanish, right? The thing that makes the shell of cockroaches and copepods in this case and so on. Can they actually degrade the chitin? Can they grow on it? Can they secrete the enzymes that degrade the chitin? can they grow on the monomers? That's the sugars that come out, the sugars out of hydrolysis of chitin, the breakdown. And we also ask this question for the dimers, not just the monomers. That doesn't matter too much. And we ask whether they can swim. And basically, the answers are yes, pretty much for all of the things that come early, and pretty much to first approximation, no for the things that come late in the community. So what I think we're seeing here is, again, this. So chitin, the first round of invaders that can degrade, grow, do whatever. Things are released. There comes the other guys that don't care about chitin. Okay? So it's that thing, I think. Okay? And I think this also, yes, it's what I wrote here. And it's also very nicely, in my opinion, visualized with this thing here. So what you have up here is the Shannon diversity. Yes. Yeah.
The maximum that you can organize this is only in taxonomic orders, but not in species, because there is a lot of difference between the A in the first row and the A in the last row. So how can you...
Difference in what, sorry?
The characteristics, like if secretase in enzymes and other things.
You mean
like
here?
This. Yes.
Yeah.
So in the first row we have an A, but in the...
yeah. Okay, you're talking about the taxonomy.
Yes.
Yeah. So exactly. So there are some correlations. This is it's a good point. So all those things here. So these are Alteromonas, these are Vibrios, Alteromona. They are different families, actually. And maybe even classes. So really different things. And then here I also have an Alteromonas. And here I have an Alteromonas that is not following the same rules. And then here I have other things that these R's, these are Rhodobacters. We never see them appear early. But for the rest, there is no correlation. They can really move to first, later, they can degrade, they cannot degrade. There is no association with taxonomy.
Okay.
Only these R's, these Rhodobacters, as far as I have seen, have any association. So this is exactly related to the point I want to make with this whole story, is that where are the units here? We are measuring the wrong things. It just turns out, now if I know this, if I had a way to magically group these things into one variable, then this would be much simpler. Everything would be very simple. You know? And maybe all these stories about, how complex microbial communities are and so on, this is just because we're measuring the wrong things. This is my rant, but I think that's, yeah. So I think this simplicity, you can see it kind of here. Like this is the Shannon diversity. So just the diversity of species here, as calculated by the Shannon index. Initially, it increases, sorry. That's natural because you have a process of colonization, right? So the particle has nothing, now things are raining in from the environment. They get more diverse. And then later, diversity decreases. Who knows why? Who can guess why that is? No? Any ideas? You should be able to figure this out. So imagine this particle has nothing. I put it in the seawater. Microbes start coming in, sort of as random. So diversity obviously goes up. And then diversity goes down. Huh? You say competition for space? Okay. Any other ideas? Yeah. Maybe, yeah. Essentially, it's fine. Thank you. But essentially, I mean, it's what you would expect. I mean, it's the random zoo thing. You put in a bunch of guys there. Some will grow, some will not grow. It's just like what you expect. I mean, essentially, this is a really strong filter. Is that you're asking things to grow on this thing that's actually pretty hard, chitin. A very small fraction of the organisms in seawater are able to do this. And then once they manage to do, then diversity decreases, because it's not richness, not number of things, it's the Shannon index. It's like if you have something really abundant and the rest are not abundant, diversity goes down. So they become very uneven. Right? So that makes sense, going down. And in fact, now the other part of the plot, before I continue, this is, now, you remember I told you at the beginning that you can sequence all the DNA and reconstruct the genomes and all? So we did that. And then you can look at the enzymes that we know are associated with the degradation. And we can look at the frequency of those enzymes in the genome, and it goes up and reaches a maximum pretty much at the same point where diversity is minimum. So it's what I'm saying. You get a selection. It's a selection, basically, for the organisms that can degrade, which is what you would expect. Now, what I did not expect is the fact that now this frequency of enzymes declines and diversity goes back up, because that's the second round of colonization of this S2 in my sketch there. The other type of species.
Sorry, I didn't understand how you define the Shannon diversity coefficient.
Say again?
How you define the Shannon diversity
This is just
coefficient?
P log P, sum of P log P, yeah. Where P is the frequency of the species. Yeah, sorry, it's just the Shannon index. It's something people do, but. Yeah, yeah. And in the meantime, many of these things that were able to win the competition initially, at least until 48 hours, they left the particle. So that's the surprising thing also, you know, that this thing, well, we know they left, but in this it's not totally obvious, but they became less abundant. Yeah. So, okay, so that's it. And I guess this next slide is a little bit of, oh, man, this is just my description of what I said in figures, but we did an experiment. This is just sort of beating the dead horse at this point, but we took these organisms, we had them in culture, so we took these organisms that cannot grow on chitin and asked why do they, or can they grow on chitin when the other guy is there, just to confirm that this was really this process of facilitation along the trophic chain. The answer is yes. So the figure may not be super clear, but this is just two of these organisms. One of these R's, the Rhodobacter, and another guy that is a Flebobacterium, in case you care. And this is how much they grow on chitin alone. This is the fold growth from the concentration of cells you put in at the beginning to the concentration of cells you measure, I think, after a week on chitin, because it takes a while to degrade. And this is the log. So this is one. This guy basically did not change, did not grow. This guy started to die. But when you put it in with any of these organisms that are the first species, this thing here that degrades, when you put it together with any of those, then they grow. So which is also interesting in a way that they did not care. They don't care who is the first degrader. They are indifferent. And so they just can grow with anything, okay? Which again, just reinforcing my whole point here is that if they don't care who's the first species, then why do we need to count them separately, yeah? So I think this is a compelling argument that we should be able to group them and just look at the dynamics of, for example, these two groups and something like that.
Aren't we reinventing the trophic levels at this point?
Yeah. What do you mean? But what do you mean reinventing?
Reinventing in the sense that these form like plants, right? Like it doesn't really matter if the plants are eaten or they produce something that other things eat. Like these are the producers, right?
Yes.
And then it's going to be consumers and then secondary consumers. So we already have like what we want to do at this point, which is a full chain like of trophic levels.
So I don't understand. Oh, sorry. The question is, I don't know if I can repeat it. It's like maybe you can.
The question is, do we already have a name for what you're looking for? And that name is trophic level.
Oh, no, exactly. No, yes. That I agree completely. We know this type of things. What I meant is not what is the class of phenomenon, but in any particular system, can you tell me who is in what trophic level? That's what I want to know. I know we had to do all these experiments and now maybe in an ad hoc way, I can tell you these things should be grouped together, those things should be grouped together, but there is no way from the data to be able to say, these are your trophic levels, you know, because if we had that, then instead of when people look at the gut microbiome, when they are like puzzled by the 30,000 different species, and they find 10, if we're able to do that, then that's a huge difference. And now you can make models and you can make a lot of things. But we cannot make models of any of these because it's like hundreds, 10,000 of things. So no, That's the problem that I am. Good. Yes. Is it time for? How are we doing with time? I'm completely lost.
It's like
For me, it would be a good time to take a break. Maybe it's a good time, I think, to, yeah.
Let's thank Otto for this first part of the lecture. We have the break now, and then we come back for the questions. 10 minutes, like always, please.
No,
quería probarlo a mitad de la presentación porque el vídeo mejor no tocarlo.
No, just explain it perché. Però non hai mai detto nessuno, tu lo dici
presentar 15 minutos. Va a presentar 15 minutos más y luego vienen las preguntas.
Iba a preguntar si es que tenían preguntas, pero...
Bien.
No lo entendí. Ah, ya. Okay. All
Okay.
right. So we're back. So far, any questions? Do you guys have any questions so far? Maybe this could be a good time, because I'm going to switch a little bit gears, not too much. No? No questions? Okay, good. So I'll start to get into the topic that I actually care about, which has, you probably, I mean, I've said it many times, this question of what are the functional units, how to find them, and so on. And I will only talk, I will talk about how we think we can deal with this experimentally, but the real issue is how to look at any data set and how to find those things. And for that, we don't have good answers. So oh, I'm sorry, I had this here. So this is the problem. What are functional groups? This is what people call, in the field, at least microbiology, functional groups. And there's a big confusion about what those things should be, and the statistics are done in different ways, and it's a mess. But this is, I think, the initial observation that sort of started the thinking of people. So those things up there in the first row of this figure, this is a Nature paper on the human microbiome. And so these are just, each column here is a different sample. So now, there's a lot of money going into this human microbiome projects, and therefore there are thousands of samples of individuals, their feces basically, right? And it's sequenced through the methods I described vaguely before, and now you have an estimation of the species content in the system using this 16S that I also mentioned and that Jacopo mentioned. And with that, in this particular case, things are clustered at a really high taxonomic level. This is phyla, which is like really, really high taxonomic levels. Like for microbes, they're so diverse that you can do that. And so the colors indicate different phyla. That's all you need to know. And so the samples, the different humans, the different stool samples are the columns. These are, well, sorry, I made a mistake. They're not, these are the stool samples. And the others are different parts of the body. Okay, it's indicated down here. For example, the mucosa in the mouth, and so on. But we're only looking now at the upper part, okay? And so you can see that there are a lot of differences. Okay, that's the statement. They're ordered in this way so that they look more similar to, I mean, one sample is more similar to the next one and so on. And that's why you see them like this. So this is the stool sample. Stools is basically the large intestine. There is a lot of this thing called bacteroidetes that are the ones that degrade the starch and the complex. I mean, with all this Brazilian food and South American food that is packed with starch, mandioca, and what is it, banana de terra, and all these things, these bugs should be very happy. That's what they are doing. And then in other parts of the body, there are other type of organisms and whatever. But there's a huge variation, So here, there are many more of these firmicutes that are the fermenters, and much less of these bacteroidetes that are the degraders. And here there are more of the other guys, and so on. And so a lot of differences, right? That's a statement, one statement. But then, what people did was, let's look at the whole genomes. Again, what I was telling you, that you can do either the 16S, that thing that determines what is a species, operationally, and you can look at the genes. And when you look at the genes, you can also group them into functional pathways. For example, central carbohydrate metabolism, okay? So those are a bunch of genes that are related to the degradation of sugars and the utilization of sugars in the cell and so on. And you can count them as well, and so on. Other things, they're amino acid metabolism and whatnot. The ribosome, for example. Apparently, when you look at those things, then you normalize by the abundance of these metabolic classes. This now looks, as you can see, much more stable. Okay, that's the observation. You see that all over the place. Doesn't matter what the system is, you always see these things, which again reinforces the idea that we are measuring the wrong things when we are looking at this type of data. Okay, it's just that That's what we are taught to do in a way, count species. That's what technology forces us to do, count species. But and we don't have any other way to measure functions, which is what we really care about. This is what you have to do post hoc. Okay, but this thing doesn't come without problems, you know. And right now, I can tell you right now, you know, you will be surprised, but people don't do null models of these things. So what I'm trying to say here is that this class, central carbohydrate metabolism, is a large number of genes, very large number of genes, large number of pathways, all type of things. So this is a really large fraction of all the sequencing reads. So naturally, it should have a lower variance, you know, than an individual species. Nobody checks these things. So here you have a very small number of things, order of 20, 30, 40, something like that. Here you have 30,000, 100,000. And so of course they're more variable, but nobody checks these things to start with. So that's one of the problems. For example, it's just very early stages, very few quantitation on this type of things. Fine. And also another problem is that it's really not easy to get this type of data. I mean, yet this is much more expensive than this thing here. For example, Jacopo was talking about this type of data, like what's up there with the ASVs, the species, for data sets that are all over the world. There are really tons of data, so you can do what he did. But this type of data is much less accessible. It's much harder to sequence, much more expensive to sequence full genomes and infer metabolic pathways. And one more problem is that only like 40% of the genes have annotations. So actually, most of the genes, you cannot assign them into these functional groups. Okay, so that's a big problem statement. And I can tell you right away, we don't have good solutions. I will discuss next time some ideas about how to solve that problem, at least for our particular system that I just described before the break, and also in the ocean microbiome, what has been done. And we can discuss that. I'll tell you now how we try to solve this problem in an indirect way, really, for the type of system that I was talking about. It's really not an effort to solve this problem, but it's related to it. So turns out that the students in the lab had the idea of repeating the same experiment I just talked about, but with different resources, with different types of organic matter. So now I'm again talking about this complex organic matter, which before was chitin. Now they did it with alginate, which is obviously present in algal cell walls and algal organic matter, agarose and carrageenan, which are other type of polysaccharides that are in the ocean. At the beginning, I thought this is a pretty bad idea. Why are we doing this again? But turned out to be kind of insightful, I think. So anyway, so they actually learned how to manufacture these particles in the lab, so we can make them. The black things there are the magnetic powder that we put in. And again, just like before, this green thing is the bacteria attached to the particles. In this one, you cannot see it very well, but there are a lot of bacteria attached. Okay, so we can play the same game. I already told you how these things work. And we see the same story. No, but this is actually somewhat surprising, I think, that the dynamics. Okay, sorry. Let me make sure that I explain this well. So the taxa again are on the rows, time is on the columns. Okay, but one important thing, if you're paying attention here, is that the taxa here is not necessarily the same one here. They are not aligned across the different successions for the different substrates, right? They are just ordered. Obviously, we have ordered them by time. At the time they picked, they reach a maximum density. So you can see this nice trend, but they're ordered independently for each of these heat maps. Okay, but otherwise, what I wanted to say is that these dynamics of succession look pretty similar. You know, it's not like you can look at it and say, oh, this was alginate. They look very similar. So that's kind of nice. I'm not sure what it means. Oh, now this works. Because you changed it, yes. Right, so that's what it says, same kinetics, but not necessarily the same taxa. It's just the same mode of assembly, but the species can be different. And so now the next question is, okay, what if we were to align these heat maps so that we have the same species on the same row? What would this look like? Okay, and this is what it looks like. Okay, so what are we seeing here? Basically, we're seeing that there, again, sorry, this just to be clear, these columns with the colors are the different substrates. There are 4 substrates: chitin, alginate, agarose, and carrageenan. And so these are species that became abundant because this light yellow towards white color indicates high abundance. Became abundant in all the systems, regardless of the substrate, okay? And it's actually like half of the things that we can detect have very similar dynamics, very similar abundance, regardless of the substrate. They didn't care what we gave them to eat, okay? Then there are the other ones that are a bit more specific, like these guys here are enriched in alginate. These guys here are enriched in carrageenan, and so on. And then you can see these white lines, so they are separated. This is just clustering. We just did very stupid, R. I think it's just clustering that thing based on these patterns, and we find the best number of clusters using these statistical techniques that are also very simple. The silhouette, and turns out it gives you 5 clusters. This is the best number of clusters. 5 clusters. One of the things that don't care what the substrate is, and 1 cluster per substrate. So it's kind of nice because we can simplify again this very diverse system into these groups, which, as you can imagine, based on what I'm trying to preach here, is that maybe these are like functional units that we can use to simplify the system. What's here is honestly not too important. We came up with this niche breadth score, which is basically indicating what you can see already by eye here. All these things here that are present everywhere and have similar dynamics have a high niche breadth, which is simply, actually, it's again like a Shannon entropy across the 4 classes, which are the 4 substrates. So if this is high, it means that they were even across all of them. If this is low, it means that they were very uneven. Yeah, that's not too important right now. And these are just examples. These are the even guys that come from here. These are the stars that indicate where they are. And they have very similar correlated dynamics across the different communities, again, regardless of the substrate we fed them in. And these guys obviously are the specific ones. Like this guy became abundant in agarose. This became abundant at the beginning in alginate, but not in the other substrates. They are specific ones. Okay, just so the question that we wanted to ask with this is, okay, what would happen now? Okay, fine. We get some simplified version of this community structure as a function of the different substrates. Apparently, there is a group of things that don't care about a substrate, and there's other things that are very specific. Nothing in between. That's the important part, actually. And I guess that's why the only reason why I wanted to show this is because this histogram is bimodal. So it tends to be like either you're specific or you're unspecific. Maybe I can tell you right away, these things, by the way, that are unspecific, that don't care about the substrate, what do you think they are? If the world was as simple as this picture, which ones are they? Here or here? Who says here? Who says here? Okay, a lot of people don't. Most people don't say anything, but yeah, it will be this because these guys are interacting directly with the substrate, so they should care. These guys are downstream, so well, it's not obvious that they shouldn't care, but I can tell you because we have done the analysis that they don't care because the R2, the things that are the intermediates, are very simple molecules, very simple intermediates of metabolism that anybody can take. They are like acetate, amino acids, so a bunch of organic acids. Organisms tend to be not very specific to these things. At least that's my idea. But anyway, we know what's being transmitted, and it's this type of molecules. Good. So what I wanted to do is now, what if we put 2 substrates instead of 1? But how much time do you have? How much time do you have? That's it?
No, because we started late. But if we want to have some questions, then
I have to finish now.
well, now.
I'll finish in 2 minutes. 2 minutes. Now-ish. Okay. So what happens if we put two substrates in these things? Okay, what do you think happens if we put two substrates instead of one? And what I mean happens, I mean the outcomes, what are going to be the outcomes? They're going to be, I mean, what would be the simplest outcome that you would expect in terms of what's present in the community? I'm sorry? More coexistence, yes. But in terms of this thing here, what would it look like if I had another column that had, for example, carrageenan and alginate, the blue and the red? What would that column look like? You will have the first,
The first block and the second block you're together.
Yes, you will have the first one, and you will have, if it is the red column and the blue column, you have the first one, and you have the second column and the third column. It's basically what you're saying. The simplest expectation is this is just additive, that interactions don't matter. Yeah, and just in the interest of time, yes, you're right. So that's what we see. So which is it's the simplest one, but it's not surprising. It's not obvious that what you should get is the simplest answer. And so basically, what we think happens is that you have these trophic systems, pretty much trophic systems, and then if you just combine them, there are things that are substrate independent, there are things that are substrate dependent, and then when you add them, if you have a trophic system, you should expect more or less a linear combination. What would break this if you have a lot of loops, a lot of lateral connections, then things go south. But otherwise, this is what you expect. And I can tell you pretty much, maybe in the interest of time, I tell you this is what we see. I can show you the picture. That is basically what you said. These are there. Right? But we quantify this, look at the dynamics. The dynamics are really like linear combinations of the dynamics of the two communities. So it's pretty much an additive system, right? Okay.
Questions?
Hello, thank you I find this experiment really cool. And I was wondering did you or could you actually also track the dynamics of the substrate through time? Because then maybe you could try to answer these sort of networks, also looking from the sort of frequency of the substrates. Types.
That's a good question. Not in these experiments. In other experiments in the lab with more control, we have done that. Yes. I mean, the main thing I can say is that obviously the polymer, the organic complex organic matter, is consumed. But what's interesting is that that's one substrate, but very complex. And then what comes out, this R2, is a whole explosion of chemical diversity, all type of molecules that I don't even know what they do. And so that fits this next level. But that's what's interesting to me, is like this is really the increase in entropy that people talked about for many years in community assembly and so on. You can see it very clearly there in terms of the chemistry. Yeah.
More questions?
There's
thank you for the talk. It's possible to, between those groups, that you can have very less of one, the first one that needs to attach first to the particles, and then, I don't know, redundancy to supply the function by the other group, or that dynamics can happen?
I'm sorry, again, could you repeat?
If you have one of the groups, it's possible to have one of these functional groups from the.
I mean, you have to have at least two.
At least these
the
two
first
groups.
one and the second one.
Yes. Okay.
But yeah. Okay. I mean, you don't have to have. You have to have at least the first one. It just so happens in the environment, the other ones are always there, ready to come and invade.
Yeah. So the importance to attach first to the particle are these ones.
Yeah.
Okay.
Yeah.
More questions?
Thanks. My question is about the first part of the lecture. When you say that the species doesn't matter, just the main explanation was because of random. But if you look at the food web that you made upon the functional groups, maybe that food web is shared among all the microcosms, and that structure is what make the behavior the same across time. I'm speaking about when the richness decrease in time. Maybe that behavior is explained because of a same structure in the network.
So I guess you could also get this result of what you put in is what you put out in proportion. If you had only like a trophic system with no lateral connections, no loops, because there are basically no interactions. I mean, there are interactions, but they're only facilitating. And so I guess that would work as well. I think you're right in that sense. Does that make sense? Like if it was just this is the nodes are going to be species, okay? And so on. And then what you put in is what you put out, I think, right?
Or I think there's one difference here, like you have this meta community always present. Right? In the other experiment, what you put in. In the other experiment, what you put in, you cannot add more species later, right?
Yeah.
Here you have this water column, so these species has just to be somewhere, and then, because you're shaking them, at some point they can come in, right? So that I think it's a slight difference compared to the previous example.
Yeah. There's no migration. Yeah, exactly. That's the thing. Yeah.
So more questions? Maybe a quick one, short one. Long one as well. People are super hungry, I guess. So, well, let's thank Otto again for this second talk. And very, very quickly, short announcement. There is another change in the program that is actually removing the change. So the roundtable is going to be today in the afternoon as it was planned in the beginning. So today the schedule keeps as it was initially. We'll come back at 2:15 for Priyanka's talk, and then we have the roundtable. We come back, anyway, we have Bill Bialec's talk today, so there is no rush to go back. So 2:30, so we have more time to have lunch, Priyanka's talk, and then the roundtable after.
So those of you who went drinking last night and are hungover and sleepy, you'd better be alert during my lecture or I'll do something regrettable.
So have a lot of coffee after lunch.
Okay.